Hello, so today we'll be talking about mitosis and meiosis. Now, mitosis begins with the interphase that is composed of gap 1, the S phase, and gap 2. In all three phases, the cell grows by producing proteins and cytoplasmic organelles such as mitochondria, and endoplasmic reticulum, or ER. Duplication of the chromosomes, crucial for the eventual division of the cell, occurs entirely in the S phase. So, the duplication of the 46 chromosomes occurs here. In G1, the cellular contents, excluding the chromosomes, are duplicated because in the S phase, that's when the chromosomes are duplicated. So the cellular contents, such as the organelles, are all duplicated here. So cellular contents would include organelles. In G2, the cell double-checks the duplicated chromosomes for error, making sure any needed repairs. So if there was an error that was found in G2, the cell would stop going forward into the next phase and would begin making repairs or stop the process entirely. The next phase is mitosis, also called the M phase, but we'll get into that later on. And after mitosis, it goes into cytokinesis, where the new cell, this original cell and the new cell both split into two identical daughter cells. Now let's talk about mitosis. So mitosis, let me write that here, begins with the interphase, which is the G1S and G2 phase. It then goes into prophase, where the chromosomes that were duplicated in the S phase begin to pair up. After that, it goes into metaphase, where the chromosomes line up in the middle of the cell. So this would be what it would look like. In anaphase, the sister chromatids are pulled apart at the centromere. So this is what that would look like. Right here. This is a centromere that is pulling the sister chromatids apart to their respective sides. And now the sister chromatids are only half. It's only half now because they're being pulled apart. In telophase, which occurs after anaphase, the cell is pinched in the middle. So the chromatids that were pulled apart to their respective sides are each on their respective sides of the cell, which will look like this. But now there begins to have a pinch in the middle, where they're starting to split into their two separate cells. And this pinching is called cytokinesis. At the end of mitosis, the product is two identical daughter cells that have identical genome properties. Next, we'll talk about meiosis. Meiosis is quite similar in some ways to mitosis, but instead of having only one part, mitosis has two. It begins the same way as mitosis, but in meiosis, there begins a crossing over of the chromosomes. So they will start swapping their genetic properties. So they'll start mixing up. In prophase 1, crossing over continues, and the paired chromosomes condense. So this is in prophase 1, they start condensing. In metaphase 1, the homologous chromosomes line up in a double line. So instead of in prophase, in metaphase, and mitosis, which is right here, we'll look at that. The sister chromatids in metaphase, they were in one single line, as you can see right here, one single line. But in meiosis, they line up in two double lines. So that would look like this. But they're still lined up in the middle as with mitosis. Now in anaphase, they begin to be pulled apart, as with mitosis. They begin being pulled apart into their respective cells. This also, in telophase 1, these chromosomes are still, they have been pulled into their separate cells, and their cells have been split into, and their cells. Now the chromosomes begin to line up in single haploid cells. This differs from mitosis because in mitosis, they were diploid, and they had both sets of they were diploid instead of haploid cells. Now these haploid cells also begin to divide, go through telophase 2, where they also divide as in mitosis, but except at the end of meiosis 2, there will be 4 haploid daughter cells. This differs from mitosis because in mitosis there were 2 diploid daughter cells. And also they do not contain identical genetic information. They are somewhat varied because of the crossing over that occurred in meiosis 1. So let's refresh. Okay. So mitosis begins with interphase, which is a part of G1, G2, and S phase. In G1, the cellular contents, such as the organelles, are duplicated. In the S phase, the 46 chromosomes are duplicated. In G2, the cell double-checks the duplicated chromosomes for any error and makes any needed repairs. If there is an error that is found, then it will stop going on to the next phase and will start making repairs. The next phase is called mitosis. It is also called the M phase, but I'll review that again. And it ends with cytokinesis, where the 2 cells are split, and now you have 2 diploid daughter cells. Mitosis begins